everyone. Welcome to learning activity number one, which is entitled The Proper Use of the Brightfield Compound Microscope in Bacteriology. And I will be giving you a prelim discussion about it. As for the specific objectives, each one of you is expected to explain the theoretical principles of Brightfield microscopy, appreciate the importance of using oil when viewing under oil immersion objective, identify the different parts of a bright field compound microscope, explain the functions of the different parts of the bright field compound microscope, list the ways of properly carrying and maintaining the bright field compound microscope, appreciate the importance of correct manipulation of a bright field compound microscope, accurately visualize prepared slides using the different objectives of the bright field compound microscope, draw the structures, visualize using the oil immersion objective, and accurately calculate the total magnification of the focused specimen. As for the expected level of competency, each one of you can correctly handle and manipulate the bright field compound microscope with ease and confidence. These are the different materials that we will be needing for this activity. We have the bright field compound microscope, immersion oil, pens, paper, and our prepared slides. Before getting ourselves involved into the microscope, let us have a short introduction about bacteriology. By definition, bacteriology is a branch of science that deals with the study of bacteria. Bacteriology is a branch of microbiology in which bacteriologists study and learn more about the various characteristics, which involves the structure, genetics, biochemistry, ecology, etc., of the various bacteria, as well as the mechanism through which they cause diseases in humans and animals. Let us define a bacteria. Bacteria are minute organisms which are too small to be seen with the naked eye. In order for us to visualize these minute organisms, one must use a good compound microscope, and the simplest type of a compound microscope is the bright field compound microscope, which is the main topic of our learning activity number one. A microscope is defined as a device for magnifying objectives that are too small to be seen with the naked eye. Now let us discuss the reason behind why it is coined or termed as a bright field compound microscope. The word compound from the bright field compound microscope means that a specimen is positioned properly on the stage of a microscope and it is illuminated by a light source which will be magnified by a two-lens system. The image formed by the objective lens will be magnified again by the ocular lens system. Therefore, the magnification is compounded. While the term bright field refers to the fact that magnified objects appear as dark objects against a dark background, a sufficient contrast must exist between the magnified object and the bright field background for the objects to be visible. And so that is why it is termed as a bright field compound microscope. Okay, now, have you ever wondered how come does the microscope can allow us to see or view very minute things such as bacteria? Let's discuss how this optical system magnified those small images. The path of light of the specimen going to the eye. So the magnification is achieved when the light rays from an illuminator or light source passes through a condenser, which has lenses that directs the light rays through the specimen. And then the light rays then pass into the objective lenses. The lenses closest to the specimen. Then the image of the specimen is magnified again by the ocular lens or the eyepiece. And that's why you can view those images even of the smallest things. After learning the path of light, let us discuss the importance of using immersion oil. In order to preserve the direction of light rays at the highest magnification, immersion oil is placed between the glass slides in the oil immersion objective lens. Oil immersion microscopy increases the refractive index of the specimen when used properly. With limited disadvantages, slides prepared with immersion oil techniques work best under higher magnification, where oils increase refraction despite short focal lengths. The immersion oil has the same refractive index as the glass slide, so the oil becomes part of the optics of the glass. When the immersion oil is used, the light rays do not refract when as they enter the air from the slide. Therefore, the use of immersion oil improves the resolving power of the lenses. If oil is not used with an oil immersion objective lens, the image becomes fuzzy or with poor resolution. This image shows the difference when performing microscopy with immersion oil and without immersion oil. In figure A, you can see that the refracted light rays are lost to the lens, while in figure B, oil immersion microscopy diffraction is minimized as light bends the same as it passes through the layers of glass and oil. This is different from other mediums such as water or glycol, where the refractive index changes each time the light hits the glass and water, potentially decreasing the image integrity. This is an image of a Brightfield compound microscope, and it is important to know the functions of each part. We will only be discussing some of its parts, therefore, it will be your task and your responsibility to research for the various parts that will not be discussed in our pre-lab discussion. A compound bright field microscope consists of a structural and optical components. Now let's proceed to discuss about the structural components. So please don't forget these three structural components. We have the head or the body, the base, and the arm. The head or the body, it houses the optical parts in the upper part of the microscope. The base is found at the bottom of the microscope, which supports the microscope and houses the illuminator, or it is basically what the microscope stands on. The arm, it connects to the base and supports the microscope head. It is also used to carry the microscope. Now let's proceed to the optical components of our microscope. We have the eyepiece or ocular. The eyepieces are the lenses at the top that the viewer looks through, or they are usually 10 times or 15 times. It remagnifies the image formed by the objective lens. Typically, the standard eyepieces have a magnifying power of 10 times. The eyepiece tube, it is where the eyepieces are dropped in. Also, they connect the eyepieces to the objective lenses. Next, we have our objective lenses. These are primary lenses that magnifies the microscope. Usually, you will find 3 or 4 objective lenses on a microscope. The most common ones are 4 times or the shortest lens, 10 times, 40 times, and 100 times, which is the longest lens. The scanner objective is often marked with 4 times magnification. The low power objective or the LPO is often marked with 10 times magnification. It is usually shorter than the other two objectives and it usually forms the general outline or the wider portion of the object. We also have our high power objective or our HPO. It is often marked with 40 times magnification. It is longer than the LPO and it forms a bigger image of the object in focus. In most cases, it is used to enlarge specimens that are so small under LPO. The high power objectives are spring loaded. Spring loaded objective lenses will retract if the objective lens hits a slide, which will prevent damage to both the lens and the slide. We also have our oil immersion objective or the OIO. It is often marked with 100 times magnification. It has the highest degree of magnification and it is used to examine stained smear preparations of microorganisms using immersion oil as the medium. We also have our nosepiece, or the revolving nosepiece. It is a circular structure where the different objective lenses are screwed. To change the magnification power, you are just going to simply rotate the turret. We have our coarse adjustment knob. It is a bigger wheel used to adjust the LPO in focusing. It is also used for initial focusing of the specimen. While the fine adjustment knob, it is a smaller wheel used for delicate and final focusing of the specimen when using the HPO and OIO. It is also used to make the specimen more vivid. Next component or part is our stage. The stage is a flat platform that supports the slides. We also have our stage clips. Stage clips holds the slides in place. If your microscope has a mechanical stage, the slide is controlled by turning two knobs instead of having to move it manually. One knob moves the slide left and right, the other moves it forward and backward. And we also have our illuminator. It is a steady light source that shines up through the slide. Mirrors are sometimes used in lieu of a built-in light. If your microscope has a mirror, it is used to reflect light from an external light source up through the bottom of the stage. And the illuminator is usually located at the base of the microscope. We also have the condenser. Condenser lenses focus the light that shines up through the slide and are useful for obtaining sharp images at magnifications of 400 times and above. If the maximum power of your microscope is 400 times, a stage mounted 0.65 Na or greater condenser is ideal since it gives you a greater clarity without having to be focused separately. However, if your microscope goes to 1000 times or above, focusable condenser lens with an Na of 1.25 or greater is needed. We also have our iris diaphragm. The diaphragm or the iris is located under the stage and it is an apparatus that can be adjusted to vary the intensity and size of the cone of light that is projected through the slide. As there is no set rule on which setting to use for a particular power, The setting depends on the transparency of the specimen and the degree of contrast you desire in your image. And we also have your condenser focus knob. It moves the condenser up or down to control the lighting focus on the specimen. In order to maximize the function and use of the Brightfield compound microscope, proper care of the microscope is very essential. The following are the general guidelines in handling a Brightfield compound microscope. Get the microscope from the stockroom by taking the arm with one hand and supporting the instrument at the base with the other hand. Next is to place the microscope at least 6 inches from the edge of the laboratory table with the microscope arm facing it in order to avoid from falling out. Then, you have to wipe all the lenses with lens paper before and after use. If the oil immersion lens is sticky, moisten a piece of lens paper with 95% alcohol to wipe it clean. Never use tissue or cloth when cleaning the objective lens or oculars. Only use special lens paper or wipes for cleaning the lenses. Microscope lenses can easily be scratched and should be treated with great care. Never use sharp instruments or anything abrasive on the microscope lenses. Then you must keep the stage clean and dry. And lastly, do not tilt the microscope. Instead, adjust the position of your chair so that you can comfortably use the instrument. Tilting leads to distort wet mounts or objects covered with oil. Failure to clean the lens will result in the formation of gummy residue, which reduces the efficiency of the lens. Now that you have learned on the proper care of the microscope, I will be teaching you on the correct manipulation of the Brightfield compound microscope. First, you have to place the specimen slide on the stage and secure it with the stage clips. Arrange the portion of the slide to be examined over the central opening in the stage. Next is you have to rotate the LPO into place under the body tube. You will hear a click sound when it is correctly in place. After hearing the click sound, you may now raise the condenser as high as it goes, and then rotate the coarse adjustment knob clockwise to bring down the LPO close to but not touching the slide until the specimen is seen through the eyepiece. And then regulate the intensity of light by opening or closing the iris diaphragm. After using the iris diaphragm, you may now sharpen the focus by turning the fine adjustment knob and then turn the LPO to HPO without elevating the body tube. Notice that it will be almost in focus because most microscopes are parfocal. Little adjustments with fine adjustment knob will only be needed to clearly view the object in focus. After having a clear view on the high power objective, now to focus on the oil immersion lens, swing the OIO halfway towards the specimen in focus, and then place a drop of immersion oil into the specimen. Swing the OIO in place. Using the coarse adjustment, bring the objective down until it touches the oil. Find the object by gently turning the coarse adjustment knob upwards. Sharpen the focus with a fine adjustment knob. Now that you have done viewing under LPO, HPO, and OIO, you may compare the relative sizes from those objectives. And then after using the microscope, clean the stage and the lenses after each use before returning the microscope to the stockroom. Now I am going to teach you on how to calculate the total magnification. So this is computed using the formula total magnification is equal to the magnifying power of the eyepiece multiplied by the magnifying power of the objective used. The number or the total magnification, it tells the operator of how many times the focused specimen is magnified. Okay, let us have an example. So the magnifying power of the eyepiece is 10, and then the magnifying power of the objective used is equal to 100, which is our oil immersion objective. Therefore, to solve for it, total magnification is equal to the magnifying power of the eyepiece multiplied by the magnifying power of the objective used, or 10 times 100, that is equal to 1000 times. Therefore, the specimen in focus is magnified 1000 times its actual size using the oil immersion objective. Okay, now I want you to remember these terms. First is parfocal. Parfocal refers to objectives that can be changed with minimal or no refocusing. When you adjust your microscope from one magnification to another, it is nice when the lenses remain in focus the entire time. This is possible when a microscope has parfocal objectives. While resolving power or resolution, it is the ability to distinguish objects that are close together. The better the resolving power of the microscope, the closer together the two objects can be and still be seen as separate. Now let us differentiate the terms magnification and total magnification. When we talk about magnification of a microscope, it describes the increase in apparent size of an object compared with its actual size. Therefore, an object magnified 10 times appears 10 times larger than it really is. While total magnification, it is the product of the ocular lens magnification and the objective lens magnification. Now that you have learned the basic principles and theories regarding the microscope, let us proceed to the microscope troubleshooting. First, my microscope will not turn on. Possible reasons are your microscope is not plugged in. How are you going to resolve it? Check to see if your microscope is plugged in. And then, check to see if the bulbs are installed correctly. You may have to install or reinstall the microscope bulb. If the bulbs are installed, check to make sure they are not loose, which sometimes happens during shipment. The next thing is to check the rheostat, which controls the light intensity on the side of the microscope. Or you can check the fuse, because many users omit to turn up the light upon using the microscope. The next problem would be I cannot see anything through my microscope. So things you're going to do is that you have to make sure that you have removed the protective coverings such as the lens covers, ensure that the eyepieces are installed, and check that the light is turned on. The next problem would be my microscope is out of focus. Here are the things that you're going to do. So the height of the microscope condenser may be set too high or too low. So you're gonna adjust it. Make sure your microscope objective lenses are screwed into the body of the microscope all the way. And then your microscope cover slip may be too thick. This table shows the commonly encountered problems upon using the microscope. and the possible causes and what are the possible solutions in order to troubleshoot those problems. The field is dim, so it can be the condenser may be too low. So the solution is to raise the condenser. And then the condenser iris may be closed. Therefore, you have to open the diaphragm. The next problem would be dark shadows in the field which move when eyepiece is moved. Possible causes is that the eyepiece may be dirty, so you have to clean the eyepiece, and then the surface of the eyepiece may be scratched. Therefore, a new eyepiece may be needed. The next problem will be the image is not clear. There are a lot of possible reasons of this kind of problem. First is that the smeared portion of the slide may be upside down. The solution is to turn the slide over. The next one is that there may be an air bubble in the oil. Therefore, you have to move the 100 times lens from side to side. The oil used may be of poor quality. Therefore, you must use only good quality immersion oil. And then lastly, there may be dirt in the lens, so you have to clean the lenses. And the last commonly encountered problem in microscopy is that the image through LPO is not clear. So the possible causes are the following. There may be oil in the lens, so you have to clean the lens. The second one is that there may be a dust on the upper surface of the lens. Therefore, you have to clean the lenses. And then the lens may be broken. Therefore, you must need a new lens. And that would be all for our learning activity number 1. I hope that you have fun in reviewing the proper use of the Brightfield compound microscope. And thank you for listening, everyone. God bless you and more power.